Hello, everyone, and welcome to our webinar. It's entitled, Taking Whole Genome Sequencing into Primetime Clinical Practice. This is the first webinar from Clinical Omics, a digital publication created last year by the publishers of Genetic Engineering and Biotechnology News. Clinical Omics has become the go-to resource for clinicians, including pathologists, clinical lab directors, technologists, and others interested in the transfer of omic technologies into the clinic. Clinical Omics provides a roadmap that can be used to improve patient diagnosis and treatment. Last March, JAMA published a paper on the clinical interpretation and implications of whole genome sequencing. It led to a lot of discussion and debate on the use of whole genome sequencing in the clinic. Today's webinar will demonstrate how whole genome sequencing is on its way to transforming medicine. Our panelists, Dr. David Smith, Dr. Jason Park, and James O'Leary, will discuss the benefits of whole genome and other next-gen sequencing techniques and point out the barriers that need to be overcome before they can become more effective clinical tools. I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotech News, and I also wear the same hat for Clinical Omics. I'm delighted to be serving as the moderator for this, our first Clinical Omics webinar. During the webinar, we invite you to tweet about anything you find interesting using the hashtag capital W, capital G, capital S, capital R, EADY, capital FOR, capital P, RIME, capital TIME. So it's WGSR ready for prime time. And you'll find the widget for tweeting at the bottom of the screen. At the end of the webinar, after the panelists make their presentations, there will be a question and answer segment. Type your question at any time during the webinar into the Q&A box on the left side of your console. Then hit submit. The panel will try to answer as many questions as possible. So if everyone's ready, let's get going. Dr. David Smith will be our first presenter. David is Professor of Laboratory Medicine and Pathology at the Mayo Clinic. He is also the chairman of the Technology Assessment Group at the Mayo Center for Individualized Medicine. David, all yours.
Thank you, John. So I'm going to start off and first talk a little bit about next-generation sequencing. NGS are a variety of technologies that are based upon massively parallel sequencing. The whole goal is to bring the cost of whole genome sequencing, WGS, from hundreds of millions of dollars per genome down to affordable levels. The very first next-gen sequencing platform was developed by 454, but then was subsequently developed by Illumina, and other companies to develop platforms include Solid, Ion Torrent, Complete Genomics, or CGI, and Pacific Biosciences. And then there are other platforms that are under development. The platform of NextGen by Illumina is illustrated on this slide, and on the left is the actual flow cell that can run 8 different samples, or the same sample in each lane. And the key behind next-gen sequencing by Illumina is bridge amplification of your template. So DNA molecules are taken and fragmented. Oligonucleotides are placed at the ends of those molecules, and then they're denatured and flowed across the flow cell. And attached on the bottom of the flow cell are oligonucleotides which are complementary to the oligonucleotides which were placed on the outside of the DNA fragments. The DNA fragments can bind to their complementary oligonucleotides, and the other end, which actually is then complementary to other oligonucleotides, can bend over, and what you have generally formed is basically a bridge and a template, and this can be copied, and then by doing this for a series of maybe 30 to 40 cycles, what you end up doing is making multiple copies of each individual template, so you end up with clusters, and on the whole flow cell, you have clusters of clusters. This is then placed into the sequencing machine, and the sequencing platform does sequencing by synthesis. Some of the key features of this are that the 4 nucleotides are each blocked. They have a different fluorescent color, and when they are added to this, they bind to the individual cluster where the base that they're complementary to exists. And then subsequent to that, a photograph is taken, and you can see what color has actually been incorporated. Because the nucleotides are blocked, only a single nucleotide can be added. Subsequent to that, the photograph is taken, the blocking groups with the fluorescence color are removed, and this is ready for a second cycle. And then each cycle, one base at a time, sequencing by synthesis is done. They are then lined up together, and through this, one can generate a sequence at each of the individual clusters. There are multiple platforms that Illumina has And it starts with the very first one, which is a MiSeq, and the MiSeq can run a small number of 15 gigabases of sequencing. The next seq can run 120 gigabases. The third machine, the HiSeq 2500, can run up to 1,000 gigabases, and the fourth machine, the HiSeq 10X, can run up to 1,800 gigabases. Yesterday, 3 additional Illumina machines were announced, and they look like to be filling the space between the HiSeq 2500 and the HiSeq 10X. Now, in terms of the evolution of the instrument performance, this next slide shows the output on the original genome analyzer, which started in 2006, producing less than a gigabase of sequence data, through its life cycle to the very end, where it was capable of generating almost 50 gigabases of sequence per run. And in blue, you see the output of the HiSeq 2000. But this shows the actual output now brought out to 2014, where in a period from 2006 to 2014, the output on this machine increased up to 1,000-fold. And this brought the cost of genome sequencing and all other next-generation sequencing down considerably in price. Now, there are limitations on Illumina sequencing. The first one is the lack of viable competitors. Illumina sequencing controls some 90 to 95% of the sequencing space, and without viable competitors, Illumina is not under pressure to improve in terms of output, improve in terms of customer service. The other issue in question is there a limited room for considerable further improvements in output? The output of over 1,000-fold increase over the past 6 to 8 years has been amazing, but it does raise the question if they're going to be able to continue that. The other thing is that Illumina sells machines and sequencing reagents, but then you're left on your own in terms of bioinformatics and genome assembly. For example, the HiSeq X10 can produce an entire genome of sequence for $1,000, but how much does it cost to assemble and interpret that? And that could be considerably more. Complete Genomics is another platform which offers a completely different type of service. It's a short-read DNA sequencer, but it is not commercially available. Complete Genomics is a place where you send your samples to them, and they do the sequencing and assemble it for you. This was purchased last year by BGI, but they offer an integrated sequence and assembly for one cost that is competitive. They also have a long fragment read technology that still uses short read technology, but with dilutions of the starting material can produce phase-accurate mapped complex genomes. And this is presumably going to be available sometime, hopefully by the end of 2015. And their third-generation CGI platform with this offers the capability of producing genome sequences for less than $1,000. And on this platform, the sequences that are generated are also assembled with BGI pipelines. And there seems to be considerable room for improvement to this platform so that over time, this can offer even greater output for lower prices. And the next slide shows their customized service for cancer sequencing, where you send them your tumor DNA and your matched normal DNA, and the first step is to confirm that the samples match. And then the second thing is to do the sequencing, which is now shown on the bottom here, the short read sequencing. And with that is then taken and you can assemble your normal genome, which is compared against a reference genome, and you can look for things within the normal genome. But the importance of this is then you can then examine the tumor genome, and the tumor genome can first be compared against the reference genome, but it can also be compared against the matched normal genome. And with this, you can see all the types of comprehensive differences, somatic differences, small variants, copy number variation, lesser allele frequency, and other things. And with this, you have the ability to very quickly compare a cancer genome to a matched normal genome. The limitations with CGI whole genome sequencing, one of the major ones is they have the machines and you have to send your samples to them. But a second one is that you don't have the capability to take unassembled data files and play with them as you do with the other platforms. You simply get the assembled sequence from them and then you can look at it. A third platform is the Ion Torrent, where the chip is the machine. And this shows the actual chip. It looks very similar because it's built almost the exact same way that a computer chip is built. And on the next slide shows how that chip is actually aligned. The Ion Torrent machine basically has a large number of very small pH wells that can measure change in pH. And it turns out when a base is incorporated, there is the release of a hydrogen atom, so that what happens is there's a change in the pH of each well when a base is added at that position. The limitations with the ion Torrent are, first, that it uses emulsion PCR as the way to amplify DNA fragments. The bases themselves are unblocked, and this is very similar to the 454 platform, and this results in a large problem when you have long homopolymer stretches because all of the bases are added, and it's difficult to discern differences between 7 bases, 8 bases, 14 bases, 15 bases, etc. In addition, another limitation is that they're considerably behind Illumina in terms of output, and thus they're not yet ready for whole genome sequencing, at least yet. However, this platform is very good for small gene panels because it uses much less sample input DNA than Illumina. Now, in terms of the uses of next-gen sequencing, there are many uses. The subject of this webinar is when is whole genome sequencing going to be ready for the clinic, but it turns out next-gen sequencing can be used for many things in addition to whole genome sequencing. There is WGS or whole genome sequencing, but you can also use it for whole exome sequencing, for small exome, small gene panels, for RNA sequencing or RNAseq, or for methylation sequencing. In terms of the strengths of doing whole genome sequencing, it provides complete genome information and organization. It can provide complete phased genome information so you can tell which is on the maternal and which is on the paternal chromosomes if done properly. You can discern mutations and other alterations throughout the genome, translocations, changes in copy number, and information about viruses if they are present. However, there are weaknesses for whole genome sequencing. One is that it does not provide any information about what is actively being transcribed. The second and the largest problem is that it poses considerable bioinformatic challenges. How does one assemble that? Does one assemble it against the reference genome? What are the various ways to do that, and how much does it cost? The second thing is the total cost of whole genome sequencing. How much does it cost to do the sequencing? How much does it cost to assemble it together? In addition, it raises the question, does it actually provide too much information? And it's too difficult to look through all that information to try to find out what are the important alterations and what are the causative ones versus ones that are not. But another additional problem is that the sequence that is generated is a considerable amount of sequence, and it costs some money to store it. So all of these are weaknesses to whole genome sequencing, which is why other things are sometimes considered. For example, whole exome sequencing. In contrast to whole genome sequencing, it requires considerably less sequencing than whole genome sequencing. It's only 38.5 to 40 megabases of sequencing instead of the 6 gigabases that is required for whole genome sequencing. Whole exome sequencing is cheaper than whole genome sequencing, at least currently, and it can produce a sequence depth for the average base of 200 plus very easily. Analysis is considerably easier than whole genome sequencing, and you're storing much less information than whole genome sequencing, which raises the question of why not focus on the important part of the genome and instead of worrying about the entire genome. The weaknesses are that whole exome sequencing only examines the exome. There are considerably more important sequences than just the exome, for example, non-coding regions, for example. The capture does not get every base. This is actually going to be discussed in a little bit more detail in the next talk. There's limited information about alterations out of the exome, and it's difficult to determine copy number changes, certainly outside of the exome, and viruses will only be detected if they're actually integrated into the exome. Otherwise, they will not be seen. The next type of thing would be a small exome or a sub-exome or a small gene panel. The strength is it's even less sequencing than whole exome sequencing, and again raises the question of why not focus specifically on what you're interested in. It's even easier in terms of the bioinformatic analysis because less sequence is being analyzed, and in certain instances can be cheaper. However, there are weaknesses. One is that you can only find what you put on the panels, so it's less comprehensive than whole genome or whole exome sequencing. Whole exome sequencing actually offers a single universal panel, whereas the small gene panels means that you need to devise specific panels for each application, and depending on how many things are on your small gene panel, it can almost be as expensive as whole exome sequencing, but providing considerably less sequence and information. There's no information about copy number changes outside of what you capture, and certainly, like all these others, no information about transcriptional changes. Now, RNAseq has a variety of different strengths. One is that you can obtain complete poly A plus information or complete transcriptome information, depending on if you want to just look at the polyadenylated transcripts or if you want to look at all the transcripts. And the only difference between the two is for the polyadenylated transcripts, you oligo DT prime, and for the other, you first remove the ribosomal sequences and then sequence all the remaining RNAs. It identifies all transcribed sequences and viral transcripts if present. It produces a digital message abundance, which is very important because it has the accuracy over a wide range of message abundances. It easily detects rare messages, totally depending upon how much sequencing you do. It can detect transcript isoforms. It can detect novel transcript fusions. And for the most abundantly transcripts, it also can detect mutations. And it very much complements whole genome sequencing as well as whole exome sequencing. But there are weaknesses to the RNA-seq, and one is that it's difficult to determine transcript isoforms if there are a considerably large number of such isoforms. And then it raises the question of how much sequencing is enough, and this totally depends on what you're looking for. If you're looking for extremely low abundant transcripts and you want to look at isoforms, you need to do a lot of sequencing. But there's also questions about how to integrate the different RNA-seq data together, transcript abundance, transcript isoforms. And more importantly, how does one integrate RNA-seq data with other NGS applications, certainly like whole genome sequencing and whole exome sequencing? Now, methylation sequencing is extremely important. It has a number of strengths because it provides important information that complements whole genome sequencing or whole exome sequencing. It's also extremely complementary with RNA sequencing. And methylation is more directly affected by environmental factors. So by determining the status of methylation throughout the genome, you find out additional information about gene regulation. But there's a number of important weaknesses. One is that the actual way that methylation does control gene expression is not well understood. Is it all CPG islands at the 5' end of the genes, or is it random CPGs throughout that are more important? This has not been resolved. In addition, there are many methylation sites throughout the genome, and there really are not very good programs to analyze the actual effects of methylation, and making sense of this type of data is thus extremely difficult. Now, these are the different ways and different applications of next-gen sequencing. How can they be applied clinically? Well, the next slide starts to talk about this. Well, whole genome sequencing is a way to comprehensively look at the genome, but when would this be appropriate? When is it necessary and important to look at this much information and make sense out of it? Whole exome sequencing, which is much simpler than whole genome sequencing, can be used. And one of the areas where this could be used very quickly is in solving the diagnostic odyssey. You have a child that's born with some abnormality and you don't know what's wrong with that child, and rather doing it gene by gene, one sequences the complete exome to try to find out what's wrong. Now, small gene panels also have clinical applications because they can be used to replace Sanger sequencing-based tests. And for many types of tests, you just want to look at a small panel of genes to see if they're mutated. And RNA sequencing could also have potential clinical uses, but it raises the question of when. When do you need RNA sequencing, and when clinically would this information help you to make better decisions for your patient? In terms of the clinical next-gen sequencing at Mayo, in 2012, Mayo started looking into developing next-gen sequencing for clinical applications. And the very first test that went live just about a year after that was a hereditary colorectal cancer panel, which was a test of 22 genes involved in hereditary colorectal cancer. Since then, Mayo has begun to adopt next-gen sequencing in a variety of things. So there's a variety of different small gene panels, one for the hereditary colorectal cancer, but there's also a cardiovascular panel, a neurodegenerative panel, and others are going live. In terms of cancer treatment and care, there's a 50-gene cancer panel, which is live, which looks at mutations in the exons of 50 genes. In terms of whole exome sequencing, when and for what would whole exome sequencing be used? It turns out at the Mayo Clinic, for research purposes, there's a considerable amount of whole exome sequencing being done. But in terms of using whole exome sequencing in clinical practice, it's not being done yet at the Mayo Clinic. And whole genome sequencing is not even close clinically at Mayo, but it is being explored in research purposes. Now, obviously, there's another issue about next-gen sequencing, and that is not in us performing next-gen sequencing, but in us actually seeing patients who already have their genome sequenced showing up at the Mayo, and the reality that this has already occurred. And anecdotally, I've heard this from other places and other centers as well. So in terms of what is the lowest hanging fruit and where is the first area that at Mayo Clinic, but at also other places, we're going to see next-gen sequencing really take off, it has to be cancer. The cost of treatments is so expensive, and any improvements, for example, stratifying patients, could result in huge savings, so thus this would justify the cost of the sequencing up front. Will this be reimbursable? I think it's going to be very easy to prove the cost savings with this strategy, so that what most likely in the next 1 to 2 years, we're going to see small gene panels and RNA-seq as the way that many cancers are going to be looked at. And this is the beginning of next-gen sequencing starting to be done and utilized at the Mayo Clinic and elsewhere. Thank you.
And thank you, David, for that superb and thorough overview on what whole genome sequencing and other NGS methods can bring to the clinic and what improvements still need to be made to be applied clinically. Thank you. I'd like to remind our audience that you can tweet about this webinar using the hashtag capital W, capital G, capital S, capital R, EADY, capital FOR, capital PRIME, capital TIME, WGS ready for prime time. The webinar widget is located at the bottom of your screen. And you can also send in a question for our Q&A session at the end of the webinar by using the Q&A box on the left side of your screen. Our second presentation will be given by Dr. Jason Park. He's an assistant professor in the Department of Pathology at the University of Texas Southwestern Medical Center. He also works at the Advanced Diagnostics Lab at the Children's Medical Center in Dallas. Jason, the floor is all yours.
Thank you, John. Again, my name is Jason Park, and I'm at the University of Texas Southwestern Medical Center. Today, I'm going to be discussing challenges in clinical genomic testing. Broadly, we will cover two areas today. First will be a review of what's been published and discussed regarding clinical genomics, and we will cover the areas of clinical exome sequencing, whole genome sequencing, and gene panels. The second part of the talk will cover a particular quality issue which will be important for all these types of clinical genomic tests. As a brief overview, when we consider the human genome, there are approximately 23,000 genes. Of these, only about 3,600 have known mutations that are associated with disease. An alternative way of looking at that is that there are about 5,000 diseases with a known genetic alteration. Overall, genetic diseases are not a common problem in the population. It is estimated that only about 2% of the U.S. population has genetic diseases. However, if we look at special populations, such as pediatrics, we know that about 70% of hospitalized pediatric patients have a genetic etiology of their disease. Some of this has been covered by our previous speaker, but as a basic overview, we should have some context of what it means to have genomic testing. There are a variety of genetic testing technologies. Some are focused, as indicated on this slide, at the chromosomes, so broad changes. And historically, we've looked at karyotypes, which can detect deletions that are greater than 2.5 million base pairs. And then more recently, in the last 10 years, we've started to do microarray technologies, which can now detect below 5 kilobases. And still a mainstay of testing deletions or translocations is the use of fluorescent in situ hybridization, or FISH analysis. The type of technology that this forum is discussing is DNA sequencing. And here is just a brief historical overview to give us context for what we're doing now. If we think back to the mainstay of genetic testing in the 70s and 80s, it was Sanger sequencing by using radioactive technology. And on the far left in black and white, we see a radiography pattern of giving individual discrete bases for a gene that's being sequenced. In the middle there, we see Sanger fluorescent technology, which was the mainstay of the Human Genome Project. And important to remember, to give us context, that we could, in a single instrument run, we could sequence about 1 million base pairs. In the last 10, 15 years, we've started to adopt so-called next-generation sequencing technologies and technologies that look at single molecules. And so in the picture under there, you see the fluorescent pattern from an Illumina sequencing machine. And in this type of instrument, certainly you can have greater than 100 billion base pairs in a single instrument run now. So this type of sequencing technology, looking at billions of base pairs sequenced in a day, has enabled clinical next-generation sequencing to occur throughout the world. In the United States, starting in about 2011, 2 laboratories began to offer clinical exome testing. In this slide here, you see the several labs that I've mentioned and the types of exome sequencing methods that they use, but there are more than 16 clinical exome labs in the United States alone. When we look at whole genome sequencing in the clinical setting, there are several different labs, and I've listed 2 of the ones that offer this service now. Whenever we talk about clinical genomics or exome sequencing or whole genome sequencing, it's important to remember the differences between exome and whole genome sequencing. So if we look at a handful of parameters shown on this table, the first one I've listed is the target size. So this is what we're trying to examine. This is the goal for each of these tests. And in exome sequencing, we're trying to look at 30 million base pairs, or 0.03 gigabases. In comparison, the goal in whole genome sequencing is looking at 3 gigabases. The amount of sequencing that needs to be done typically to achieve a meaningful resolution of each of those targets is about 7 gigabases for an exome and 90 gigabases for a whole genome. If we skip down to the minimum depth at base, what I mean there is what is the number of reads that needs to occur at a single base position in order for us to be able to detect a variant. With exome sequencing technology, this number is usually about a depth of 20 reads. In whole genome sequencing, this is a depth of about 10 reads. Finally, what do we actually get? So we're trying to target an exome about 30 million bases. How many of those do we actually get meaningful data from? And depending on the studies that have been published and the different reports from clinical labs, this is about 90 to 95% of coding bases. In comparison, through a variety of studies with clinical whole genome sequencing, the numbers are slightly higher at 95 to 98% of coding bases. And I've listed here some of the charges that I'm familiar with for each of these services in the clinical setting. Now, there's been a tremendous amount of work in clinical exome over the last couple of years, and this has resulted in a number of landmark studies which were published in 2014. So this table summarizes some of the early studies going back to 2012, as well as the flurry of studies that occurred in 2014. So overall, where are we right now? Well, in these studies, there are about 3,000 patients that we have data on, and we know that their diagnostic yield, so the ability to arrive at a genetic diagnosis for these patients, is about 16 to 60%, which is a very, very high number compared to other single gene sequencing tests. But important caveats are that this is predominantly a patient population that is pediatric, that mostly have neurologic disease. So whether or not this is generally applicable to other types of diseases and other patient populations is not certain yet. In comparison, clinical whole genome sequencing is relatively new. And there were 2 studies in 2014 looking at clinical applications, and I've summarized them here. So in comparison to exome sequencing, there are only 56 patients that have been published on, and the diagnostic yield is not higher. It's about 24 to 50%, so it's a significant amount of overlap to exome sequencing. Importantly, many of the pathogenic variants which were found by whole genome sequencing are coding variants. And so presumably these would have been detected by an exome test. As an example of that, the next slide takes an in-depth look at one of the clinical whole genome studies. And this study found the genetic cause in 21 cases. In 16 of these 21 cases, we could presume that the same findings could have been done by current exome or array technologies. So what I've shown here is 2 different tables. On the left is a table of each of the genes which had a single nucleotide variant detected by whole genome sequencing. But in that left table, we also see a column labeled Baylor MGL. And so this is a clinical exome lab that has put available to the public its ability to cover each of these genes. And so in red, I've listed 2 genes that have imperfect coverage.